Hello everyone, this is lecture number 46. In previous 2 or 3 lecture, so we are looking for unit number 5, and you know that unit which is divided into two parts. First part was on-site sanitation treatment system, in which that already we learn about septic tank, its design, and what are the package system, then treatment of working principle of package system, advantages, disadvantages. Then lastly, in previous lecture, we seen as some important a new introductory term as MBR, MBBR, and FMBR. Now this was of related to the first part of unit number 5. Now in the second part is related to the anaerobic digesters. Okay, so second part we start here, that is anaerobic digestion or digester. So in today's lecture, we will see here introductions of anaerobic digester, then principle of anaerobic digestions, the stage of digestion, and what are the factors governing anaerobic digestions. So the fourth topics or fourth term which will be see in today's lecture. So first, as a anaerobic digestion. So meaning of anaerobic digestion is that here anaerobic is the process by which organic matter such as animal or food waste is broken down to produce biogas and the biofertilizers. This process happens in the absence of oxygen in a sealed oxygen-free tank called as anaerobic digester. So anaerobic digester is the one of the assembly, one of the unit which we are, so you know that there are the different types of organic matter that came from that produced by animal or food waste is broken down into the produce. That means there is the organic matter in that particular unit and which is broken down and produce the biogas or biofertilizer. This process happening in the absence of oxygen in that particular unit, and which is a free oxygen of that tank. That is, it is a airtight tank, which is called as anaerobic digester. Mostly anaerobic digester is produce the biogas of organic matter. Just remember one thing, if they will ask the question, what you mean by anaerobic digestion? It is one type of unit which produce the biogas of organic matter, and if that organic matter by the animals or the foods. Is that clear? This is about the anaerobic digestion, and it's mainly focused on it's free from the oxygen. So you can see here such type of unit is called as anaerobic digestion. Okay, so which can be produced the biomass. Okay, so a digester is a large container. So you can see there's a digester having a large container in this shape, in which substances are treated with heat or enzyme to promote decomposition or extract the essential component. Okay, just a minute. Okay, okay. In the this particular types of a digester, what happening is there exactly substance is treated, okay, with the heat or enzyme, and it promote the decomposition, degradation of organic matter, and through which once they degrade that matter, at that time extract the essential component such as gas, which is called as a biogas. You can see here, okay, so this is a stabilized solid which is in the form of organic matter in that particular tank or particular assembly, which were what they can be treated as with the heat of enzyme. At that time active biomass, so supernant and skim layer activate here, and at that time once the degradation or the de uh, the word is decomposition of that particular organic matter, at that time does some extract essential component that is nothing but as biogas, which is came into the in this direction, and this is the inlet of that organic matter, and this is the outlet process of that entering matter of particular units. Okay, this is called as suspended growth anaerobic digesters. Okay, and after decomposition of the all the matters, organic matter, it can be a withdrawal in the directions or through the bottom of that unit, that is solid withdrawals. So anaerobic digestion mainly focus on what? Anaerobic digestion is naturally occurring process by decomposition of and decay. So it is a natural process Then here the organic matter is broken down into the simpler chemical compounds under the anaerobic conditions. So organic matter which is simply broken down in the simpler chemical compounds, and once they are broken down in simple chemical compound, it can be easily decomposed, and that time you can extract some component in form of the gas. So anaerobic digestion is digestion of organic material. The process focus is on what exactly process. You can see here anaerobic digestion is the focus on what digestion of organic material. Then in absence of oxygen, main focus on what it working on the in absence of oxygen, then it can forming end product of methane or carbon dioxide. Okay, so what the end product of that particular digester is what to produce two product byproduct after decomposition degradation of the different compound of organic matter, and its the end product it will be produce the methane and the carbon dioxide, and that methane and carbon dioxide is nothing but as the biogas. ओके सो जुन्या काळात जर तुम्ही बघितलं कदाचित प्रत्येक शेतकरी जी मोठी शेतकरी होते त्यांच्याकडे खूप गाई म्हशी सारखे पदार्थ होते तिथे बायोगॅस असेंब्ली होती बहुतेकाच्या तिथे ते युनिट प्रोव्हाइड केलं होतं अँड दॅट इज अ व्हॉट anaerobic digestion. जर तुम्ही जुन्या शेतकऱ्यांकडे जर बघितलं तर मेनी ऑफ अ फार्मर विच आर ऑलरेडी अवेलेबल इन देअर फार्म्स विच इज कॉल्ड anaerobic digestion. Okay, so an anaerobic digestion is naturally occur process by decomposition and the decay. So just discuss about it. So mainly what are the principle of anaerobic digestion? We can see here anaerobic digestion is a process through the which bacteria break down organic matter. So mainly how to break down that matter, which help you the break down bacteria break down organic matter, then such as manure, then without oxygen. Okay, so bacteria which are help to break down the organic matter in the different compound, but it without oxygen. As bacteria work, they generate biogas. Okay, so just I told you the bad bacteria can be generated the biogas. The non-methane component of the biogas are removed so the methane can be used to energy sources. And once the produce of methane gas, at that time non-methane component can be reduced, can be removed, and then methane can be used as a energy sources. So here are the some stages of anaerobic digestion. That is what hydrolysis, acidogenesis, and the methodogenesis. So you can see here the particular organic substrate that is protein, carbohydrates, and the fat. So you know that organic matter, this such type of substrate are the available. So proteins and the carbohydrate, which is break down into amino acid and sugar, and fats, which is break down into fatty acids. Okay, so amino acids and the sugar which is came from the proteins and the carbohydrate, which is going to the what propionic acid and butyric acid, intermediates of propionic acid and butyric acids, and the amino is goes into the acetic acid, and it again goes into produce the methane gas. But as sugar which is going to the propionic acid and the butyric acid, it can be produced again acetic acid and the hydrogen by the fatty acid through. And these two term byproduct of these two acetic acid and hydrogen can be produced the methane also here. But this whole process which is go through the four stages. That stage is the hydrolysis stage, acidogenesis stages, acetogenesis stages, and the methogenesis stages. So one by one you can see here in detail. So what do you mean by hydrolysis? Hydrolysis is a large organic compound are converted into the simple monomeric compounds. Okay, what organic compound, large organic compound, it can be converted, it can be broken down into simple monomeric compound. Okay, just we are discussing about the hydrolysis process, hydrolysis stage. Okay, how to produce methane, carbon dioxide, or methane or the other gas. That is our biogas. Okay, then secondly, it is accomplished through the extracellular enzyme. Okay, for example, it completes to what extracellular enzyme. Cellulose gets converted into the starch using the cellulase. Then casein gets converted to the amino acid using the proteases. Then triglyceride get converted to the fatty acid using the lipases. Okay, so this mainly focus on these two word or two factor, that is what large organic compound are converted into the simple monomeric compounds, and second it completes to the extracellular enzymes, for example, are the given here. That is called as what? Hydrolysis stage. Then secondly, stage is that acidogenesis. In the acidogenesis stage, the soluble monomeric compound from the hydrolysis undergone fermentations. Okay, though those matter which are the soluble monomeric compound, the soluble monomeric compound which is produced by what? Large compound or large organic matter. That means a large organic matter is break down into a small monomeric compound. If that monomeric compound later to be soluble, that means in the liquid form, at that time by hydrolysis undergone fermentations. Okay, the product of obtained due to the acidogenesis are the propionic acid and the butyric acid, and which are the intermediate are the others such as acetic acid, ethanol, hydrogen, and the carbon dioxide also. It's a byproduct of the monochromic compounds. Then an example of the bacteria that bring about fermentation are the lactobacillus sp. Okay, fermentation is due to the only hydrolysis undergoes and under the process, and at that time in this stage due to the acid production is the pH fall down. Okay, in this stage pH is fall down, acid is produced here. Okay, you know that pH is a fall down, that means less than 7 it may be acidic. It is a general example. Then acidogenesis. Some of acetate is the produced through the mixed acid fermentation. Okay, so now hydrolysis process go into monochromatic liquid based soluble. At that time acid is a generated, acid is a produced here, and once the stage of acidogenesis which can be produced through the mixed acid fermentations. Okay, rest of acetate is produced through the secondary fermentation product obtained in the previous stage. Then acid formation is important for the formation of the methane, and that is a very important thing. That is acidogenesis is nothing but the acid formation, and is important for formation of the methane. Example, acidogenous bacteria, that is Acetobacter special. Okay, sp, which is called as Acetobacter special, which can be helped to the what? It is the mixed acid fermentation through which the acid formation is important to the formation of the methanes. Then last stage in the uh, aerated sorry aeration digestion, anaeration digestion actually. So in methogenesis stage, this is the last metabolic stage in anaerobic digestion. Okay, actually just I told you this is the last stage of anaerobic digestion. Methane is formed either from acetate or carbon dioxide. So generally in the chemically or chemistry, as per the chemistry knowledge, methane is formed either from the carbon dioxide, otherwise acetate acid, and the hydrogen. These three elements which can be form the methanes. The bacteria responsible for carrying out the methogenesis are the called as methogens, and are classified as what? Eutrophic or the hydrogenotrophic, that is they produce methane from the carbon dioxide or the hydrogen oxide. And acidotrophs and the acetoclastic, this convert acetate to the methane. Okay, mainly these two factor or two component or two element, that is acidotrophs and the acetoclastic, which they can convert to the acetate into the methane, and methane can be generated through the three element, that is what? Carbon dioxide, acetate, and the hydrogen. And methotrophs convert into methanol to the methanes. Okay, these acetotrophs, acetoclastic, and the methotrophs, which can be convert the methanol into the methane gas. Okay, this is about methogenesis stage, and that methane gas as used for the source energy, that is nothing but the biogas energy. Okay, this is about how to work, how to process, what are the what are the process in that anaerobic digestion in the four stage we just seen here. Now what are the factor governing aerobic digestion? The first factor, that is temperature. Mostly temperature, methane forming bacteria are activated in two temperature ranges. Okay, when methane forming bacteria can be active. So that first factor we consider here, that is the temperature. If the range of that mesophilic range from 30 to 35 degrees Celsius, at that time methane forming bacteria should be active. The thermophilic range from 50 to 60 degrees Celsius and the temperature between 40 to 50 degrees Celsius, at that time ethane forming bacteria are inhibited. Okay, so this temperature variation, it should be remembered here when a methane forming bacteria can be active. But more than 40 to 50 degrees Celsius, at that time ethane forming bacteria inhibited. Okay, but it 30 to 35, at that time it can be active in terms of mesophilic range, which is called as. Okay, so this is the first parameter or first factor which we can governing with the aerobic digestion, that is the temperature. And then second is the pH value. So if you consider the factor of pH value in aerobic digestion, so digester pH is the 7.2 or lower than that. The presence of what NH4 plus is favored. When the digester pH is greater than the 7.2, the presence of NH3, that means hydrogen, is favored. But dissolved ammonia gas or NH3 is toxic to bacteria, especially methane forming bacteria. Okay, this is about the pH value, which is related to the pH, NH, and the ammonia gases or toxic gases. Then a fourth one factor or third one factor, that is nutrients. Nutrients means what? It can be growth of the bacteria to produce the methane gas. Okay, so here we can say the nutrient as a micronutrients. So in the micronutrients, there are two types of main micronutrients. One is the nitrogens and second is the phosphorus. Okay. So in the micronutrients, methane forming bacteria possess several unique enzyme system, and the needed of several micronutrients, especially cobalt, iron, and nickel, and the sulfide is critical. Okay. So mostly the nutrient, the micronutrients are working, and the micronutrients are classified into two. That is, one is nitrogen, the second one is the phosphorus. Okay, so this is about third one factor which is governing to the aerobic digestion. Then fourth one, that is mixing. That is a very very important. Mixing enhance the digestion process by disturbing bacteria. Okay, what? Mixing enhances the digestion process by distributing bacteria, substrate, and the nutrients throughout the digester, as well as equalization of temperature. Methane forming bacteria are very sensitive to rapid mixing. If rapid mixing continuously washes out the methane forming bacteria in the effluent, this is about how to mix that the organic matter, and through which that how to generate the methane gas, all they put here. Then fourth one factor, that is seeding process. Okay, seeds an anaerobic digester with the adequate population of faculty to anaerobes. Anaerobe is nothing but as what? Absence of oxygen microbes can be growth, that microbe can be seeds. So anaerobes include the methane forming bacteria, and aerobes, that means the microbes which are having forming the bacteria, that is methane bacteria. It should be ratios as 1 as 10 of secondary sludge to the primary sludge may be used. Without seeding, it may take a few months to get the tank operational, and proper seeding will be help to attain the quick balance of the reaction. So seeding are the very important absence of the oxygen, that is called anaerobes. Or anaerobe means that those microbes, those microorganisms that can be growth without consumption of the oxygen. Okay, it can be help to the forming of the methane gas. So it was the some factor which is governing with the anaerobic digester or anaerobic digestion. Now, what are the advantages of anaerobic digestion? You can see here. It produces methane and the carbon dioxide which can be collect and they use as a fuel. Okay, this is the first advantages. Then reduce the volatile content of the sludge. Then third one, liquid more easily dewatered as liquid and the solid formed of the separate layers. So you know that organic matter broken down into different types of compound which can be helped to the liquid more and easily dewatered. The reduction in number of pathogens, that is 13. Again, here some disadvantages of anaerobic digester. It needs to constant supervision, that is very important. Then formation of explosive gases may be produced there. They're not extremely controllable. so there is no any control of that particular assembly, so that is exclusively controllable. The maintenance and cleaning is very difficult, then it is slow process. So these are some disadvantages. So this is about what in today's lecture we learn of four terms, okay, and just be seen in here. Thank you for attending it.